This is the Podophyllotoxin Photolysis presentation by Rabbi Ashok, Zuhair, Hussain, Elliot, Chen, Raghuram, Vungutur, and AnhMinh, Pham. Podophyllotoxin is an antimitotic agent which prevents mitosis in cells. It is produced by the plant species Photophyllum, peltatum, and Sinopodophyllum, hexandrum, royle. It is utilized in the chemical synthesis of chemotherapy drugs like etoposide, tenoposide, and etoposide. In our study, we mainly focus on using the prodrugs 5-dimethyl and 5-methoxy. We use them because they're very similar properties, so we can assume that they will behave similar when we analyze them in the HPLC. Our goal was to decompose these prodrugs using photolysis and then analyze the results by running them in the HPLC to see if the separation of the photophyllotoxin into two separate molecules.
Just some background about the machines that we were using. So we mentioned HPLC, which stands for High Performance Liquid Chromatography. And it's a method used for separating, identifying, quantifying numerous components in an analyte mixture or compound. And in a normal HPLC, the column is polar and the mobile phase or the solvent we use is nonpolar. And RP-HPLC stands for reverse phase HPLC, and it's a specific type of HPLC that analyzes and then separates substances based on their polarity. So in an HPLC, it's the reverse, where the column is nonpolar and the mobile phase is polar. And due to its usefulness, RP-HPLC is used for medical, manufacturing, legal, and research purposes. And just one example is RP-HPLC can be used to test samples of urine for performance enhancement drugs in high-stakes competitions. And due to its capability to separate many compounds, it's commonly used globally to verify drug purity in a variety of pharmaceuticals. So down below, we just have a breakdown of the different parts of the HPLC and the RP-HPLC. So we have the mobile phase, the solvent pump, the liquid sample, our column, and our detector for the HPLC. And then the next slide should show kind of a picture of the machines that we're using, as well as what it looks like inside the HPLC when it's injecting from the sample files.
So before we could start photolyzing the sample in the HPLC, we first needed to create a method to run in the HPLC. And for the method, we needed to find the correct gradient so that when we ran it in the HPLC, the sample would elute between 10 to 15 minutes. And to do so, we ran a sample of both prodrugs in the HPLC for 30 minutes. And in data analysis, there should be one peak. And at this peak, it shows where the solution eluted in the column. And like I said, the peak should be between 10 to 15 minutes. But if the peak comes out at either earlier or later than this range, we have to adjust the gradient and run the HPLC again. And in this slide, on the left, it shows one of the methods that we ran, where the peak appeared a bit too late. So we had to adjust the method in order for the peak to show up earlier. And for the other one, the opposite happened. The peak arrived too early. So we adjusted the method so that the peak would show up later.
Okay, so after we finished the method development, we were ready to move on to the next step of the experiment, which is to perform photolysis. Photolysis is basically a chemical reaction in which an inorganic chemical or organic chemical is broken down by photons. So what we wanted to do was perform photolysis on the podophyllotoxin, and then to verify that the podophyllotoxin actually broke down into two separate compounds, we would analyze the resulting solution in an HPLC to make sure that this has happened. So the procedure to run the photolysis was, first we would prepare a solution of phosphate buffer, then we would dissolve the Podophyllotoxin prodrug in the solution and mix it. And then we used 300 nm lamps over a period of 6 hours on the prodrugs. What this would do is this actually performs the photolysis. Then to track the photolysis, we just put it into the HPLC and then analyze the results after. We also use benzoic acid because it serves an internal standard, which basically that means it's a substance similar to the analyte, and it's added in a constant amount to the blank standards and the samples. So the methods that we used for the first prodrug, 5-dimethyl, in the end, we decided on a 0 to 70 method. And then for the second prodrug, the 5-methoxy, we used a 30 to 100 method. The purpose of these methods that it can be used in pharmaceutical industry to confirm the identity of drug substances and also see if there's any impurities in these drugs. And it could also be used in clinical diagnosis by detecting diseases through analyzing precursors and also monitoring the progression of diseases.
So for the next slide, we just have a picture of some of the samples that we used. And what we found was interesting is that every 30 minutes when we would check the sample, it would change color. So the lighter yellow is after 30 minutes in the HPLC and in the lamp. And the sample next to it, where it's darker, it was in the HPLC and lamp for 150 minutes. We're not sure as to the exact cause of the color change because nothing's showing in the HPLC results, but we just thought it was an interesting observation.
So these are our current results. This is for T0, so the first injection of the run. And as we can see, we have our base peak, which is at around 80 in this height of MAUs. And if we go to T150, so this is 150 minutes in the HPLC, the peak is now much higher at around 85 to 90, and we don't know the cause of it just yet, but we're sure to find out soon enough.
Thank you to Mr. Chen, our advisor, for introducing us to the HPLC machine and being there at lab hours in order to help with the study. We also give thanks to ASDRP for giving us the resources and opportunity to perform the study using the high-tech equipment in their labs. We also appreciate all the on-site staff that helped us during this process. Thanks for listening.
Very nice. Good job. Any questions? No questions. So I'll ask you one. What is your, how many of you are staying on for the fall? And what does the fall semester look like for research as you continue?
So I can go ahead and answer your question. So usually the photolysis process takes about 5 to 6 hours based on our kind of prediction. And due to some issues with the HPLC, we've only gone up to a certain point, about 150 minutes. And we haven't really seen much change with our HPLC results. So our hope for the next semester is to find that point in time where we'll be actually able to see the photolysis and the breakdown of the compounds and maybe figure out what's the reason for the color change as well. So that's kind of our future plans.